Today, we are going to be talking about the polymerase chain reaction, also known as PCR. PCR is a method that makes millions of copies of specific DNA segments within a span of a couple hours.
This method was first invented in 1983 by Kary Mullis. Kary Mullis worked at a biotechnology company called Cedars Corporation. At the time, Cedars Corporation was working on creating tools that could clone genes. Mullis's job was to make small samples of genetic material for other scientists to use in their experiments. That's when he came across the idea of PCR.
The first step of this process is to heat the DNA molecule to 201 to 204 degrees Fahrenheit. This separates the double helix into two separate strands. These strands can be used to make more copies of the DNA segment.
The second step is to cool the sample to 113 to 140 degrees Fahrenheit. This causes the two strands to start coming back together. The next step is to insert primers, which prevent the two strands from connecting. The primers are markers that identify which segment of the DNA is to be copied.
The sample is then heated up to 162 degrees Fahrenheit, which allows an enzyme called Talc to attach to the floating nucleotides. Talc polymerase is a heat-resistant enzyme that allows for the automatic process of PCR. Once all nucleotides are attached, one strand of DNA becomes two. This cycle is repeated to continue increasing the amount of targeted DNA.
So if you have one piece of DNA, one PCR cycle will make two copies of the target DNA segment. Each copy can make more templates, and within 30 cycles of PCR, you can have over 1 billion templates.
PCR is a type of DNA analysis that can both identify and exclude individuals as suspects in an investigation. DNA identification was first accepted in court in 1986 with a case in England. In 1986, two girls were sexually assaulted and murdered. A 17-year-old hospital employee had confessed to committing one of the two crimes. Both cases were very similar, but the suspect only confessed to one. DNA testing was used to link both crimes. While both crime scene samples matched each other, they did not match the suspect who confessed. Authorities then did a mass screening of over 1,400 people and found the suspect who had attempted to give his DNA sample to someone else.
Today, PCR has a variety of uses, including its use in the Human Genome Project. The goal of this project was to analyze, map, and sequence the entirety of human genetic code.
PCR is also used to diagnose diseases and genetic disorders, sequence and clone genes, and detect mutations and pathogens, all in clinical settings.
Its use was also applied during the COVID pandemic. During this time, there were COVID PCR tests that could identify if a person was infected with the virus. A positive match would mean that the SARS COVID primers matched the DNA in the sample, which caused amplification of the segment. This means the individual is infected. If there is a negative result, the primers do not match to the DNA in the sample, and amplification does not happen.
The most common way scientists analyze the PCR product after amplification is through the use of ergos gel electrophoresis. This separates the DNA fragments based on size and positive or negative charge. This method shows the presence and size of the PCR.
There are certain factors that can affect how specific PCR can be to an individual. These factors include the purity of the DNA template, the concentration of primers and the sequence, and the concentration of the magnesium ions used to promote the activity of the Talc enzyme.
In forensics, PCR is used for DNA profiling, which includes genetic fingerprinting, DNA typing, and DNA testing on samples found at crime scenes.
Thank you for watching this video on the polymerase chain reaction.